All right, so we are on to chapter 3, part 3. And we're going to talk about DNA. So this is one of my most favorite things to talk about. As I was reading your book, as I read every textbook that talks about the discovery of DNA, they always say Watson and Crick determined that DNA is the double helix, right? Okay. So what's really true is that before about 1952, all scientists thought that the genetic material that was inside of cells were proteins, because proteins are so much more complex than, and that is my son making noise. All right, because proteins are way more complex, right? We have 20 amino acids, whereas we're talking about DNA, we only have 4 nucleotides. So how could something simpler be the genetic code? And so it was actually a big shift in thinking in finding out that DNA was really the genetic material, because just a genetic material was enough to allow a virus to replicate. So they were able to figure that out. But who really made the discovery? It's a really good question. So the person who collected the data, the quantitative data that proves that the structure of DNA, was Rosalind Franklin. And her colleague shared her data with Watson and Crick. Now, before this, Watson and Crick were making some assumptions and trying to figure out the structure, and they had come up with some ideas, but it didn't work. And when they saw Rosalind Franklin's data, that's what allowed them to make their conclusions. But Rosalind didn't give Watson and Crick the data. So there's a thing there. And while Watson and Crick were awarded the Nobel Prize in physiology and medicine for determining the structure of DNA, the award should have also gone to Rosalind Franklin, and it would have, had she not passed away. She had died, I think it was 4 years earlier, before the Nobel Prize was awarded, and they never award Nobel Prizes posthumously. Okay, and that's my phone. Golly. Oh, so sorry about that interruption. So we were talking about Watson and Crick. And okay. And so Rosalind Franklin should have gotten the credit for that as well. And if you've ever heard Watson and Crick talk about it, they say, well, Rosalind didn't know what she was looking at. She didn't make the conclusion. Well, science doesn't occur in a vacuum, number one. And number two, you wouldn't have come to the conclusion had you not seen her data. So that's a story for a different day. So if you want to hear more about that, I'm happy to talk about that. But let's get back to some real science and not history. So when we talk about DNA and RNA, what is the difference? So in DNA, our deoxyribonucleotides do not have an OH, right? They just have a hydrogen there. And so in DNA, right here, we just have the H, but in RNA, we have both the oxygen and the hydrogen. Okay, so they're super, super similar. The only difference is at that 2' carbon, we have the OH instead of the H. So you need to know your complementary base pairs, which I'm sure you probably do. A's and T's and C's and G's, apples in the tree and cars in the garage. That's how I learned it. So the only difference in DNA and RNA is that in RNA we have no thymine. So thymine is replaced by uracil. What is the difference? They're extremely similar. The only difference between thymine is the original structure here. Uracil, all you would do is remove this group right here, and it ends up being a hydrogen in uracil. Other than that, they're exactly the same. So when you're looking at these base pairings, right, guanine's always going to pair with cytosine, adenine's always going to pair with thymine. And the reason for that are the hydrogen bonding that occur, right? We know our guanine and our adenine, these are the pyrimidines. These are the purines. purines, so guanine and adenine. So the 2 rings are the purines, and the 1 ring structures, those are the pyrimidines. M-I-D-I-N-E. And if you haven't figured this out by now, I am a terrible speller. You do not have to spell to get a PhD, by the way. So do not let that hold you back. Anyway, so you're always going to pair a purine with a pyrimidine, and it's always going to be guanine and cytosine, adenine and thymine. So when you look at guanine and cytosine base pairing, what you're going to see is that you have 3 places where hydrogen bonding occurs. If you look on the other hand at adenine and thymine, there are only 2. So when you're looking at this, if you're just looking at the hydrogen bonds, you can tell which base pairs are there. And the more hydrogen bonds there, the stronger the bond. So what's a stronger interaction? It's the guanine cytosine than the thymine adenine. The thymine adenine is weaker, guanine cytosine is stronger. Okay, so when we look at this in a double-stranded helix, because DNA is double-stranded, what we're going to see is that the 2 strands run anti-parallel. So if you look in A, if you look here, we're calling this the 5' end. Why are we calling this the 5' end? Because it's attached here at the 5' of the carbon. So the prime is actually referring to where that phosphate group is attached, right? So then we have our 3' end here. So this strand runs 5' to 3', right? And this strand runs, here's our 5', so see, here's the carbon here. So these are anti-parallel, anti-parallel. So they're complementary in that on one side, when you have a T, you're going to have an A, and anywhere where you have a G, you're going to have a C, right? See, anywhere you have C, a G, and anywhere you have a T, an A, you have a T. So they are complementary and they are anti-parallel. And if you look at how DNA replicates, right, remember the DNA will unwind, will separate, and then each strand will serve here. This will be the template strand. So here's the other template strand, and then from that template strand, you're going to synthesize a whole new strand of DNA, right? So it's a semi-conservative replication, which you should have learned in Biology 101, excuse me. All right, so here's our central dogma. How is DNA our genetic code, and how do we get from genetic code to proteins? Well, first thing that we have to do is we have to be able to replicate our DNA with high fidelity. So what was in the original strand, it needs to be almost exactly that in the new daughter strand. So we have that through DNA replication. So then we go from DNA replication, we want to get to proteins. Well, we can't go straight there. We have to go through an intermediate. Our intermediate is RNA. We have a couple of different kinds of RNA. We have mRNA. mRNA is what serves as the intermediate between DNA and proteins. So we undergo DNA transcription, we transcribe the DNA, and we go to mRNA. So that's DNA transcription. Then that mRNA is translated into a protein. Now that we have the protein, we can go and do all sorts of things. So these are our functions, right? But that's not the only mRNA that we make. We actually transcribe the DNA and make 4 other kinds of RNA. rRNA and tRNA, these are both involved in protein synthesis, right? In getting to the point where we go from mRNA to protein. But we also have miRNA, microRNA, and snRNA, small nuclear RNA. And those are involved in regulation and processing and some really interesting things. And so we're going to talk about some of those later. So what's our relationship between DNA and protein, right? So remember, we're going to start with our coding strand. Our coding strand is that template strand. So here's our coding strand right here. And so we're going, and I said that wrong, right? See if I can erase. Oh, I'm learning all this stuff, and maybe I can erase. Yay. Okay, so let me try that again. So we have our coding strand and our template strand. So what I want us to do is I want us to look, and why are we starting with different color? I don't know. Okay, is that we have here our template strand, and the template is what the mRNA is going to bind to, right? So if our template goes TAC, this is our DNA. When we make RNA, right, we're going to make the complement. So that would be A. And we're not going to make T. We would normally say T, but in RNA, what do we have? We have U. And then our complement C is G. And so that is exactly what we have here as our first mRNA codon, right? And so that would process so that this new strand of mRNA here looks exactly like the coding strand, except that the U is there in place of the T. And there's my drawing that finally popped up. All right, so now that we have this mRNA transcript, now we can go and turn that into a protein. And so that's transcription. And so we're going to take our individual codons, and in the ribosome, we're going to convert that into amino acids. And so you can look at a codon chart. I think your book has the square codon chart, but I really like the circular one. So I'll show y'all how to do that later. I don't know if y'all have seen that before. And we turn those into amino acids. Okay. Now, in biochemistry, we're going to have a lot of omes, right? So you've probably heard genome before. Genome is all of the genes inside of your entire body. But what you might not have heard before is called the transcriptome. So this is our collection of DNA transcripts. This is our mRNAs, right? And so, well, not just mRNAs, but it's all the other RNAs as well. But it's our collection of all those RNA products that are generated by transcription. Now, you got to think every single cell in the body has the same genome, but you're going to express different parts of the genome in different amounts depending on if you're a skin cell versus a heart cell, right? They have the same genome, but let me tell you, they do not have the same transcriptome. And so then that transcriptome will determine what your proteome looks like. And the proteome is our collection of all the proteins that are made by the mRNA translation, right? And so you can see how if you change the genome, then you're going to change the transcriptome. You change the transcriptome, you're going to change the proteome. So depending on where you want to change things, you can actually target different places, which is kind of cool. Oh, wait, and that's the end. Okay, end of 1.3.